Good morning, S2. Today we're going to look at microbes, disease, and the prevention of disease. So, super important cheese questions. Um, what type of cheese is made backwards? Edam. What type of cheese hides a small horse? Mascarpone. There was an explosion at a cheese factory. What was left? Only debris. What cheese do you use to attract Winnie the Pooh? Camembert. What do you call someone else's cheese? Nacho cheese. Why is Christmas the cheesiest holiday? Because of the baby cheeses. When can you not see cheese? Oh, when it's pasteurized. And what does cheese say to itself in the mirror? Halloumi. Just for your general knowledge of cheese. It's because last lesson we looked at cheese, so I thought it'd be useful to go into the wider world understanding these important facts. Let's review our yogurt. So had we made yogurt in a previous lesson, we would have added a live culture of yogurt. So that is yogurt that already exists and has living bacteria in it to a sample of milk. Over time, the milk would have turned into yogurt due to the production of what's called lactic acid by the bacteria. So yogurt is a dairy product made using a microorganism, bacteria, to make it. We're going to today look at microbes and disease. We might then further in the video look at preventing the spread of disease. You're going to understand what's meant by the term disease and name at least one disease and the type of microbe that causes it, and also symptoms and how it's spread. So the good thing about this video is you can pause it and move forward at your own speed. As a starter, we're going to look at the picture, consider how disease might be being spread. You can also suggest precautions to avoid each of these things. So pause the video and have a look. Okay, so looking at some of the answers. We have the fact that the fridge is at 10 degrees C, but the door is open. We have this man picking his nose, which is obviously a vector for disease. We have this gentleman using his bare hands to pull raw meat through a mincer. We have this lady sneezing on liver, which seems to have flies on it. We have hand feeding a dog sausages while being angry. We have a dirty towel used on the hand washing station. We have a cat. So we have animals, dead animals and predatory animals in our pie shop. We've got leakage. We've got food being left out and going nasty. There's also other things as well, like keeping meat pies in the toilet and things like that. So some microbes can cause disease. They're not as useful as our lactobacillus that made our yogurt in the previous video. What is a disease? It's anything that stops one or more parts of the body working properly. Infectious diseases are ones that are catchable, so you can catch them if a pathogen infects your body. Pathogens are just any harmful microorganism. Pathogens invade our body and reproduce, so they divide, they make more of themselves, and they try and take over our body and resources. When they reproduce, we get signs and symptoms. Symptoms even. So botulism is a rare but serious illness caused by a bacterium, Bacillus botulinum. It produces a toxin that affects your nerve. So botulism from food comes from eating foods that have been contaminated with this toxin. So if you cut yourself and you get Clostridium botulinum in the wound, then you will get botulism. Nasty. Anthrax, again, Bacillus anthrax, is caused by is the bacteria that causes the disease anthrax in humans and cattle. Anthrax is very nasty. It causes tissue decay and lots of bleeding or hemorrhaging. Cholera is caused by a bacterium. It affects the body's ability to absorb water. People die from having very watery, rice-like diarrhea. Tuberculosis caused by bacteria. You get chest pains, bad cough, and it can spread to your bones or skin even. There's lots of different types of tuberculosis. E. coli is caused by, is a bacteria, Escherichia coli, and it causes things like food poisoning, vomiting, diarrhea, and nasty stuff. You get that if you don't cook your meat properly. And coronavirus, COVID-19. There's actually lots of coronaviruses that cause a variety of things like coughs and colds and other things, as well as the more serious coronaviruses like SARS-CoV-2. It was first discovered in late 2019 in Wuhan, China, and on the 11th of March it was declared a pandemic. So Usually it's just something where a pathogenic agent is at an epidemic across multiple continents. So COVID can be asymptomatically infectious. You can have mild symptoms or you can have very severe symptoms. We currently, as of February the 25th, 2021, don't know all of the long-term effects of COVID. A lot of people who experience COVID don't have a lot of bad symptoms, but doctors working in hospitals treat it as a very serious infection and are doing a lot to prevent its spread, so we should take it seriously. Poliovirus is something that causes damage to the nervous system and paralysis. You get infected by spores from an infected person's cough or sneeze, or by contact with their infected feces. It causes lots of muscle weakness and wastage. Chickenpox, caused by a virus, gives you an itchy, scratchy rash, nausea, and tiredness. Herpes simplex, so cold sore virus, it gives you cold sores. It's one of the reasons that you should not kiss someone else's baby, however cute they are. If you have a cold sore and you give it to a baby, it can really nastily affect their feeding and also can give them lots of brain problems. So, athlete's foot, you might have all had athlete's foot or a similar yeasty fungus before. It feeds on the skin, often in between our toes, and creates itchy dead skin and pain when walking. There's lots of different fungal infections that tend to affect wet, moist, not well cleaned areas of our body, as well as things like thrush that affects the mucous membrane, or things like mouth, vagina, anus. How are microbes transmitted? So you can get them through food, so foodborne ones. We've talked about E. coli and things like salmonella. Insect-borne, so plasmodium diseases like malaria. You can get dengue, African sleeping sickness. You can get lots of different things from insects. Water, so things like cholera and other things, as you know. Coughing, so it's again the things like COVID, flus, colds, things like that. And touch. There's lots of things that you can get by touching something and then it getting into your body. Your body's natural defenses should prevent these things getting into your body through the skin. It's if you touch an open area of your body, like mouths or cuts, that they can get in. And sneezing, very similar to coughing, similar mechanism, but sneezes can usually go a bit further. If you look into your booklets, you will see there's lots of different microbes, and it wants to know how each of them are spread. Go through the video again, rewind it a bit, and have a look, and that will help you. You can also use the following slides. So, salmonella, caused by bacteria through food and drink. Tuberculosis, caused by coughs and sneezes, that's how you get it. Athlete's foot, caused by skin contact. COVID, caused by coughs and sneezes. There is some foam out transmission, etc., but mostly coughs and sneezes. Cold sores, skin contact. Hepatitis, liver issue, caused by breaks through skin. HIV, caused by breaks through the skin, you can't get it through any of these other ways. Things like influenza, so that's not how you wear a mask. Coughs and sneezes. Pretty sure at least one of those people is wearing it the wrong way up. Anyway. Ebola, so things like skin contact through food and drink and through breaks in the skin. It has a very high R number, Ebola does, so and it's quite nasty. In a close population, it burns itself out, but that's not very nice for the people in that close population. You can also get it through coughs and sneezes. So you should know the three ways microbes spread, so things like touch, things like food, coughs, sneezes. What the microbe that causes the flu is, it's a virus. What the symptoms of the flu, things like feeling quite shivery, feeling quite weak and tired. What microbes that cause food poisoning, usually bacterial, things like E. coli and salmonella. Symptoms of food poisoning, things like diarrhea, vomiting, sickness. There you go. We're now going to look at the preventing the spread of disease. So we'll look at a bit of food safety. So we should wash our hands before touching food. This prevents microbes getting into our food. We should keep food in the refrigerator. It slows microbial growth. And when reheating food, we should do so at least 63 degrees Celsius. If you look in your notes, you'll probably find there's a section that this will help you with in the food safety bit. So microbes are so small that it's very difficult to see them. So we need to wait until there's enough of them that they're visible. How can we help them multiply? So if we want to help these bacteria, how would we do that? So we've looked before at what causes decomposition to go fast. We need a good food source, and we need the right temperatures, moisture, things like that. We have a special thing in our labs called Petri dishes full of nutrient agar, not A. You can actually buy different nutrient agars, and they're good for different types of microbes. These have been specially calculated if you use a specific agar in the complex medium for bacteria to want to grow on them. So keep them in a warm incubator, and it speeds up their growth. You could have a look around your kitchen and think about how clean and safe it is. Do you use a different chopping board for cooked and uncooked food? Do you always wash your hands after touching those kind of things? Is the meat kept at the bottom of your fridge? A lot of people keep their meat right above the vegetable drawer, which is not really where you should keep your meat. But then again, why do they put the vegetable drawer at the bottom of the fridge then? Why not the top? But anyway, lots of things like that. So have a look around your kitchen and think, where would food safety rules come in here, and how could we be better? So what I would also suggest is that we think a little bit about presenting results while we're here. A big part of teaching in science is telling you the theory, the knowledge, and the content, and then you come to an exam, and a third of the marks are problem solving and data handling and drawing graphs, and you think, heavens, I don't remember doing that in class. Well, this is you doing it in class. So if you get a bit of paper, I want you to try and draw. It can be lined paper, it can be blank paper, it can be any kind of paper if you don't have grid paper. Try and draw this as a bar graph, and we will go through how you would do that properly. Okay, so bar graph, first thing you want to do is draw straight lines, and after that we will look at drawing a scale. So the numbers probably are going to go up in 1,000, so you would need 15 sections. If you have lined paper, you take 15 lines, so 0, 1, 2, 3, 4, 5, 6, 7, 8, 15,000. And then across the way you would have pizza, and then an equal space, and then chopping board, space, preparation bench, space, then a lady's hand, space, utensils. So decide what's going on the X and the Y. The thing that you decide, the thing that you are choosing, goes on the X-axis. It's what's called the independent variable. And the thing that goes on the Y-axis there would be the number of bacteria. When we're drawing a graph, copy exactly what it says in the heading as your label. There should be units. So, if it's just number of bacteria, that doesn't need units. But if it was number of bacteria and it said brackets millions, you would have to put that on your line. If you need centimeters or meters or whatever, put it in. Add the labels and units if appropriate. So on the bottom, you would put in your pizza, chopping board, preparation, etc. You see they're taking one space for the bar, then leaving an equal space. One space for a bar, equal space. So if you think about it, prisons in TV and things have bars with spaces in between them. So that's how you should do a bar graph. We'll then put our scale in, so I'd say go up in thousands. So 0, they should have a 0 here. It doesn't want to be a common 0, so you don't want it in the corner. You want it lined up with this bottom bit here, 0, and then up to 15,000, because that's the highest number. This wouldn't be a good scale because it stops at 12, so you wouldn't be able to go past preparation bench. So you work out how high up it's going to go, and then you draw a horizontal line with a ruler, and then a horizontal line down. Again, in real life this wouldn't get the mark because it doesn't go all the way to the side. Mr. Collier has a rule, that's me. Don't colour in your bar graph. If you colour in your bar graph, I'm almost certainly taking the mark off you. So don't do it. Once your teacher has checked your bar graph, you could stick it in. Obviously, I'm not going to check your bar graph unless you photograph it and submit it to the assignment. So put these in the order of growing microbes. So the first thing you would do is swab an area of interest. You would then spread it on agar jelly. You would discard the swab. You would secure it on both sides, and you would place it in the incubator. This isn't something that we necessarily need because we're not going to be doing the test in person. What are the 4 key rules for food safety? So you cook, you separate, you clean, and chill. So cook. Make sure while you're cooking, you cook at the right temperature. Don't undercook it for not long enough. And don't cook it at too low temperature. Separate, so in the fridge or preparation storage area, cooked and uncooked, chilled and unchilled should be separated. You should defrost things in a sealed container or dish. And when you're chopping, you should use different equipment and apparatus for different things. Cleaning, making sure you're cleaning with something like a general purpose kitchen cleaner, alcohol or another thing, non-ionic surfactant, to clean the area. And chilling, if you're going to eat something tomorrow, make sure you store it safely overnight. Last thing we're going to do is look at the last bit of preventing the spread of disease. Nice and quick. Why is it important to wash our hands? So, did you wash your hands? You spread germs if you don't. When to wash your hands? Before you eat, after you use the toilet, if you play with pets, after blowing your nose, before cooking, and just regularly. If every couple of hours, just go and wash your hands. It's best to wash your hands with soap and water from the sink. Alcohol gel tends to dry your hands out quite a lot and will only kill surface bacteria. If you've got a big glob of mud on your hands, it won't penetrate down. So, do we always do a good job washing our hands? What is your technique? How long do you wash them for? So, there is a specific way to wash your hands, and it might even, video might even load eventually. That would be nice, wouldn't it? Anyway, while it's loading, if it gets here in time. So what you want to do is you want to wet your hands under a tap. You want to apply soap to the middle of your palm on one side. That's fine. You then want to, this is over here. Anyway, doesn't matter. You then want to rub your palms together on both sides. You want to interlace your fingers to make sure you've got, so palm to palm. You interlace your fingers so that you're getting all of the manky stuff out from in between. You then want to make sure your fingernails and the backs of your hands are nice and clean. And quite often you want to do your wrists and ideally up to your elbows. You then rinse all that off for about 10 seconds underneath the hand tap and dry your hands. In society today, what sorts of things do we do to help with hand hygiene? So we've got alcohol hand gel and soaps that are quite good. We've got alcohol gels, hand dryers, although they are a reservoir of nasty stuff. So we're going to do an experiment to find out what is best to kill bacteria. So if you did this experiment, you would find out what is the best way to kill bacteria. So you've got an agar dish that we talked about before. You would, before you start this, you would keep the lid on, turn it upside down so that the gel layer is facing towards you, the plastic, basically the bottom of the dish is facing towards you, and you would write your initials around the outside. It's very important to do this and to do the cross before you add any liquid to the dish, or it will just leak out. After you've done that, turn it the right way up and try and keep it reasonably covered on the bench. So you lift the lid of the plate just slightly. You'll then have swabbed or collected a bacterial sample on a swab, and you will add the bacterial liquid onto the plate and flood it so that all of this plate is covered in bacteria. You'll then clean the spreader so it doesn't infect people. What you would then do is you would put washing up liquid on one side, hand sanitizer on another bit, and soap and water on this bit here. But the easiest way to do that is if you soak a little absorbent disc in washing up liquid, hand sanitizer, and soap and water, and you put 3 of these discs in the appropriate corner, what should then happen is the bacteria will try and grow. And if this soap and water is good at killing the bacteria, it won't be able to grow right beside it. So what we look for is what's called the zone of inhibition, the distance from this disc of paper that bacteria is not able to grow. What you tend to find if you do this is this. The antibiotics are very good. The alcohol hand gel is very good. but soap and water is quite often the best. So the clear zone is how close to that disk the bacteria was not able to grow. So zone C, which I don't really know what zone C was. What was zone C? They'll say soap and water is the best. So if you're filling out your notes, soap and water was the best one. It's easier for germs to spread on wet hands than dry hands, but it's also easier to clean your hands if they're wet. We never steal a petri dish completely because we want a little bit of oxygen. We have what are called obligative and facultative anaerobes that are able to grow under no oxygen conditions. They tend to be not very good for us. So we try to always, when we put the lid on a petri dish, only loosely sellotape it on either side so that you don't get nasty stuff growing. Finally, if you look through your booklet, you will see this thing, which I think is called a menorah. No, a mandala, that's what it's called. So these are some microbes here, and these are some microbes or diseases here, and I want you to put them in the mandala and see if you can work it out. The answers, if you're interested, are fungi harmful athlete's foot, fungi harmful thrush, fungi non-harmful yeast, and fungi non-harmful penicillium, where we get antibiotics from. Viruses harmful influenza, foot and mouth, and HIV, which causes acquired immune deficiency syndrome. There are helpful viruses, but we don't talk about them. Bacteria harmful salmonella, bacteria harmful MRSA, not harmful bacteria in milk or bacteria in your stomach. And then you can have a go at the numeracy task. What it is suggesting is that bacteria divide by reproducing and dividing into two. So if you start with one bacteria, after a certain period of time, they're suggesting 20 minutes, you'll have two. After 40 minutes, you would have four. The number just doubles every jump in 20 minutes. After 20 minutes, 2, 40 minutes, 4, 60 minutes, 8, 80 minutes, 16, 100 minutes, 32, 120, 64, 128, 256, 512, 1024. Nice and easy. So try and work out how many bacteria there would be after 4 hours. So 4 hours is 240 minutes. So how many jumps of 20 is that? So 240 minutes. You're just going to keep this going to 240. So you would jump to 200, 220, 240. So 512, 1024, 2048, and 4096. You would then keep doing that for another 4 hours. So again, 60 times 8 is 480 minutes. You have to keep going after 240, so you go to 260, 280, etc., etc. What you get is, after 480 minutes, we would have 16,777,216 bacteria. So this is why it's really, really important to clean things up quickly. Don't leave it until after you've had your dinner, because if you forget about it for 8 hours overnight, you've got a massive amount of bacteria growing on your surfaces and your dishes from dinner. Okay, so hopefully that made a bit of sense. I know that's quite a lot to look at today, but the video will be set and then you can work through the rest of it, your notes, at your own speed.